Hello, everyone. Welcome to today's live broadcast, 5 Steps for Publication Quality Fixed Cell Imaging the First Time, presented by Jason Kilgore. I'm Kristen Perret-Gentile of LabRoots, and I'll be your moderator for today's event. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we'll make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button in the lower left. I will now turn the presentation over to Jason Kilgore.
Hello, everyone. Thank you for attending. So I've been with Thermo Fisher Scientific now since 1999 and started out in the bio R&D group here, making a number of microscopy-based, mainly reagents and kits that researchers use all over the world. I worked there for about 8.5 years, including leading a lab group. And since then, about 2007, I moved over to technical support. And so in a given year, I talk to thousands of you, advising you on how to choose, use, and troubleshoot our products. So I'm happy to present this webinar with some of the advice I've given over the years on how to make a publication quality image. Most of the images that you'll see in this presentation are ones that I've taken over the years. Now, what do I mean by publishable image? Now, naturally, you want an image that conveys whatever phenomenon that you're studying and the results. But there are some certain guidelines that I think you generally want to have. For instance, you want to have an image that has very good signal to background, has good specificity, has a bright and photostable dye, and is able to convey the message that you need for the results that you've gotten. Here, for instance, are 2 examples of some images that at first glance seem to give you some valuable information. You see some specific labeling. For instance, on the top you see Golgi labeling on the cells. At the bottom you see a brain section with some Bergman glia. But if you look closely, you start to see some background. And compare to a cleaned up image, an image that used optimized protocols and techniques that I'll be talking about, you can see a much brighter signal that compared to the background and generally a better way to get your results. Let's talk about some of the processes that are involved in that. Now I'm going to talk mainly about microscopy. Some of this will apply to other techniques, but for this webinar I'm going to stick to microscopy and the dyes used for those, both for cells and tissues. Now we mentioned 5 steps to getting a publication quality image. Really there are 5 areas, and you can see them listed here, and I'll go through each of those as we talk through the presentation. The first area has to do with the very beginning. Just like building a house, you need a firm foundation upon which to start your assay. And that means the cell and tissue preparation at the very beginning. You know, how you culture the cells, how you fix and perm. So let's talk about those. First off, you want healthy cells. You know, never underestimate the value of the cells that you're imaging at the very beginning, right? So let's say you're culturing cells. You want to take a look at those on a transmitted light microscope to make sure that they have the right morphology, that there's no contamination, that their confluency is good. For many assays, you want perhaps a 30 to 60% confluency on a cover slip. If you're using tissue, you want to make sure the tissue morphology is good. And so you have to have proper media, serum, the nutrients, good temperature and CO2 conditions. Here at Thermo Fisher Scientific, for instance, we have our GIPCO cell culture side of the company, which sells a lot of these tools that you need, like media. So when you first begin, make sure that your cells, or at least your control cells, are as close as possible to their native physiology and metabolic state. So proper culturing conditions. And prior to beginning your assay, here's a list of some things to really consider. Examine your cells. Make sure that the pH indicator is not discolored. Phenol red is most common, and it will have a pinkish color. If the pH starts to go off, you'll see that it turns yellowish. Are the cells of the proper confluency or the proper cell number for your assay? Some assays require that the cells are totally confluent and have contact inhibition going on. Are there any signs of bacteria or fungi? Do you have any strange floaties floating around in your cells to indicate that there might be some contamination? Do the cells have proper morphology? Are they shrunken? Do they have weird extended processes, or are they how they're supposed to look? Are they blubbing? I'll show you an example of that. And are the cells well adhered to the substrate, if you were talking about cultured cells? Or for tissue, like if tissue sections, make sure they're well adhered to the slide. If they're not, they can come off, and that can cause artifacts. Also consider the basis of your assay. Do you need cells in culture? Or maybe you want to go to what is increasingly popular, a three-dimensional model. Up here in the upper right, you can see some images of spheroids, for instance, or using our algae matrix to make a true three-dimensional structure that's interconnected. And these are all considerations to think of before you begin your assay or right at the beginning of your imaging protocol. Here's some examples of some cells that aren't really in the best state to start with. In the upper left, for instance, you can see a couple of cells labeled with 2 fluorescent dyes, and the cell at the top is unhealthy. You can see what's called cell blubbing, where the plasma membrane actually extrudes out into the media, a good sign that the cell's unhealthy or maybe has the wrong osmolarity. In the upper right are some cells that are probably going through apoptosis. They don't like the medium it's in. They're starting to shrink, and you see these weird processes around the outside. And the cell on the left in that picture actually has nuclear fragmentation happening. This is a green nuclear dye called cyto13. And then at the bottom, you can see an example of some fairly confluent cells that are lifting off the cover slip that they were supposed to be adhered to. This causes some distortions that you see because you can't really focus on the cells that are not well adhered in the same focal plane as the others. So for some cell types, you need polyl-lysine or some other substrate treatment in order to allow good attachment. Healthy cells definitely make for healthy data, so have a good beginning to your process. Next, and we're talking mainly here in this webinar about fixed cell systems like antibody labeling. What does fixation mean? Well, fixation means that you're doing a chemical treatment to preserve the cells in a particular physiological state and to preserve morphology in many cases. The main way that people do that is either through a cross-linking with aldehydes or through solvent fixation. Cross-linking with formaldehyde or glutaraldehyde really preserves the structure by cross-linking proteins, the amine groups on these proteins, which creates a rigidity to the cells or tissue, preserving them in that state. Just be aware that sometimes by doing that you can mask antigen binding sites where the antibody needs to bind. And you have to do an unmasking like an antigen retrieval or protease treatments in order to reveal those sites. Also, increased autofluorescence, that is, a fluorescence native to the tissue increases upon doing the cross-linking. Glutaraldehyde has twice the cross-linking power, but it also has twice the autofluorescence. Also, you could instead use a solvent-based method like methanol fixation. This permeabilizes the cells by delipidizing and opens up the cells and the antigen binding sites. Some antibodies need a solvent-based, especially if it's a nuclear antigen. But be aware that it does not preserve the morphology as well as doing a cross-linking, like with aldehydes. You can also lose some membrane-bound components and proteins, or if you labeled the live cells with a dye that was a membrane dye, like dye IC18, it can be lost with acetone or methanol fixation. On the left, you can see an example of poor fixation where the cells actually have this fragmented appearance to them. This is cells that are labeled with a phalloidin to label the F-actin in red. On the right, you can see some cells that were properly fixed. You see a nice morphology and good actin structure to those cells. These were fixed with formaldehyde, and I have a very basic protocol there at the top, basically fixing somewhere between 2 to 4%, and usually methanol-free is the best choice. Formalin, for instance, is methanol-stabilized formaldehyde, so usually you want to use methanol-free, like EM grade, and fix for about 10 to 15 minutes in PBS or another physiological buffer at either room temperature or 37 degrees. 37 degrees is what I generally recommend, just to make sure in that last moment before fixation the cells are still in their proper physiological state. After fixation, you would then wash. Some cells that you label live are not technically fixable. Some might be retained. So I just wanted to make a point of this. If you label live and then fixed, some cells like nucleic acid stains, for instance, some dyes like nucleic acid stains do not actually cross-link in place. And a fixation won't necessarily preserve them. On the lower left, you can see some example of a tissue that was labeled with 3 colors, 3 different fluorescent dyes. And when put at a warm temperature, 37 degrees for 14 days, the dyes slowly diffuse off because they're not cross-linked in place, leaving this sort of washed-out appearance. Some dyes, however, are fixable. In the upper right, for instance, you can see 2 versions of the dye at the top of the gallery. Top left is a dye called FM464 on live cells. Top right is the FX, or fixable version of it on live cells. If you fix the cells, the FM464 on the left is lost, leading to practically no signal. But the one on the right has amine groups that are cross-linked in place by aldehydes and thus retained. Sometimes you can do a post-fixation as well to cross-link, let's say, an antibody in place so it's not lost over time as you archive the slide. Remember, proper fixation means that you preserve your target for later. Next, you'll permeabilize your cells. This is where you actually poke holes in the plasma membrane, delipidize the plasma membrane such that large molecules can enter. This is very important if you're doing intracellular targets, especially with antibodies. A typical way is to use a detergent like Triton X100, maybe at a 0.1 or 0.2% at room temperature for 5 to 10 minutes. Sometimes you'll use solvent-based like acetone to do the permeabilization. Just realize that too much permeabilization can cause loss of some antigens, and delipidation can cause loss of some dyes, especially membrane-bound dyes. Remember, permeabilization actually opens the door to those intracellular compartments. After you've permeabilized, then you go on to your blocking. And I'll spend a little bit of time talking here about the different blocking techniques. Usually when you say blocking, like for an antibody, you talk about protein blocking. If you use an antibody on tissue that's not blocked properly, that antibody can bind to proteins other than the antigen that you're targeting. And so instead, you first block with a protein blocker. In many cases, this can be bovine serum albumin, or serum, like normal goat serum that's been heat inactivated. Sometimes out there people will use others like fish skin gelatin or non-fat dry milk or commercial blockers like Blockade. I have found that bovine serum albumin, maybe at a 6%, combined with maybe a 3 to 5% serum, is a very good combination to knock down some of that non-specific binding by the antibodies. However, here we sell a component called Blockade, which you can purchase at a 1x concentration, and it's not species specific, unlike serum. So you can just wash your sample with that for 30 minutes or so to block, and then you can even use it in with your primary and secondary to block those as well. On the upper right, you can see some example images of different blocking conditions compared to each other. This is a cerebellum, mouse cerebellum section, a cryosection that's been labeled only with a secondary antibody with Alexafluor 488. With BSA or NGS alone, you can see that you get a pretty high signal. In the graph there, those are the two left columns, BSA and then NGS, normal goat serum. If you use Blockade, you see that you get even less nonspecific binding, and that's a good thing compared to autofluorescence. Protein blocking is very important in order to make sure that you get a specific, or as close to specific as possible, label. You don't want to confuse with the background. Now that's protein blocking. And remember that every assay is a little bit different. Every cell type is a little bit different in terms of the optimal blocking conditions. So I definitely recommend optimizing those on your end. Now, one type of blocking that people don't tend to realize that they need is due to dye charge. Most dyes have a negative charge on them. And because they are negatively charged, they're attracted to the positively charged areas on the cells or tissue. On cells, that's usually nuclei and mitochondria. On tissue, that can be, for instance, white matter in brain sections. There is a solution called the ImageFX signal enhancer that will block those positively charged areas, and therefore the negatively charged dyes won't bind to it. Here you can see some examples that in this image at the top of with no blocking versus normal goat serum only, and then labeled only with a secondary antibody. And you can see, although it's a fairly long exposure time, you can see a fair amount of background there, and that's those two bars in the graph. But if you use the signal enhancer solution, you can see that it knocks it down pretty much to autofluorescence there. Very important, and something most people don't think about. Here also is some examples with that signal enhancer on tissue. I mentioned brain tissue. On the upper left in this image, you can see nonspecific binding by Alexafluor 488 secondary on that highly charged white matter. But with the signal enhancer, it's knocked down to just about nothing. Very good. Remember, dye charge blocking means less nonspecific background. And then in addition to the protein blocking and dye charge blocking, there's some specialized techniques that require their own blocking steps as well. It's very common to use a streptavidin biotin amplification, and I'll talk more about amplification techniques in a minute. But if you use streptavidin biotin, you want to block the endogenous biotin in your cells or tissue first. Otherwise, you'll be labeling that as well, confusing your signal. In the image to the right here, you can see on the left some cells that have been blocked for endogenous biotin with one of our endogenous biotin blocking kits. And on the right, a cell that has not been. And the cell on the right, you can see you've gotten nonspecific binding to the endogenous biotin in the cells, mainly in the mitochondria, whereas the picture on the left that was blocked does not have that problem. And then also, if you use an enzymatic sort of detection, like with DAB or tyramide signal amplification, you want to block the endogenous enzymes in that mainly tissue. For instance, peroxidases. If you're using a horseradish peroxidase or HRP detection, like with diaminobenzidine on your tissue sections or with pyramides, you want to do a hydrogen peroxide wash to eradicate the endogenous peroxidases before you go through with your labeling. Phosphatases also, like for BSIP NBT detection, you want to reduce the endogenous phosphatases with levamisole. Always to get a specific detection. Okay, so that's blocking. One last thing I want to talk about cell preparation is autofluorescence. Cells, and particularly tissue, can have a certain degree of autofluorescence in it that can confuse your specific signal and lower your signal to background. Here's an image taken at 3 different wavelengths and then combined of a kidney section just for its native autofluorescence. And you can see in every color there's a certain degree of autofluorescence there. You don't want this, definitely. And there's a lot of different things that can lead to autofluorescence. It could be overfixation with aldehydes, especially glutaraldehyde. It could be the effects of xylene from your deparaffinization. And it could be endogenous proteins like lipofuscin, which makes those age spots in old people, right? Well, lipofuscin's inside also in tissue, especially brain. Or red blood cells are autofluorescent, or certain plant pigments. So what do you do about these autofluorescent problems? Well, first you can do a sodium borohydride wash. This is where you take sodium borohydride powder that you can buy from many different chemical companies, including the Fisher side of our company. And then you make it very freshly at 1 mg per mil in PBS, and you wash your tissue or cells with it 3 times 10 minutes. And you can see an example here of how I've knocked down some background in the red on these zebrafish paraffin sections. Sometimes people will treat their sections with Sudan black or to quench the background, or photobleaching it away with sunlight or strong UV light, but sodium borohydride is usually the best way to go. You can also amplify over the autofluorescence. Autofluorescence is usually fairly dim, but if you're trying to detect a low signal, now the signal to background is a big problem. So amplifying with a streptavidin biotin, or in this case, like I'm showing here, amplifying with a tyramide signal amplification kit. Here on the left you can see some tissue that has a pretty bad autofluorescence in the green. But after amplifying with TSA, I've been able to get a nice clean signal with the Bergman glia on these mouse cerebellum sections. You can see an exposure time of only 40 seconds on the right versus 200 milliseconds on the left. So 40 milliseconds versus 200, pretty much brighter on the right-hand side. And then finally, there's ways to use software to remove autofluorescence. For instance, you can leave one channel open. Here, for instance, I've left the red channel open, whereas the green has a few very specific spots on it that were labeled for proliferating cells with an anti-PCNA antibody on rat liver. I then took our imaging software, like our Celeste imaging software, and subtracted the red background from the green signal. And in the far lower right there, you see the result where you see just a few green spots where the specific signal is actually located. There's also special instruments out there from certain microscopy companies for spectral deconvolution, where single color dye samples are used to make a library and then of dye spectra, and then it subtracts that spectrum from your specific image. Remember, overcoming autofluorescence means more sensitivity to give you a better, more publishable image. It's all about signal to background. Okay, let's go on to step 2 here, and this is labeling your sample. Here we're talking about labeling with a primary antibody and optimizations for that. Using primary antibodies is a very common technique that's been well established, and there's lots of great references out there about the many different characteristics of using antibodies, which I won't go into here. But what are some things that you want to think about in order to get the most publishable image? First off, go with a respected company as your source for your primary antibody. And there's a lot out there that you can get from colleagues that they've made themselves in their lab, and that's fine. But generally, I'd say go with a company because they've really validated their antibodies well and have QC, quality control methods in place to make sure they're the best quality, like we do. Also, make sure that your antibody is relatively new and within its stability period and has been stored as we recommend. For instance, at 4 degrees for short term, sometimes frozen at -20. Is the antibody validated for immunocytochemistry or immunohistochemistry? You want to make sure your technique matches the antibody. There's a lot of antibodies out there, for instance, that have only been validated for flow cytometry or Westerns, so you want to check that with your supplier. Very importantly, consider the host species. Do you want a mouse primary? Do you want a rabbit? Or whatever species they come from. You want to make sure it's not the same as your tissue that you're labeling, or you're going to label endogenous IgGs in your tissue. And you want to match it with your secondary antibody, and I'll talk about secondaries in a moment. Finally, do you go with a direct conjugate that has a dye already bound to it, or go with an unconjugated and then detect it with a secondary antibody? For microscopy, we generally go with a secondary antibody detection in order to get the best signal to background. Direct conjugates are very common in flow cytometry where the instruments are more sensitive, but sometimes you can use them on cells. Tissue usually has too much background and you need a further amplification. Here's a web resource listed here where you can find an antibody selection tool on our websites. Be aware also that we will also be providing a PDF of this presentation, and you can go to that PDF to find these URLs and our resources that I'm recommending. Second, every primary must be optimized separately. There's a lot of protocols out there, and I can't tell you how many times I talk to customers who are using a protocol that they've just always used with every antibody. And I'm here to tell you, you definitely don't want to do that because every antibody is a little bit different. There's even differences between lot to lot variability from a supplier. So you definitely want to optimize the fixation time and which fixative you want to use and the time for it. Very much the concentration or dilution of your antibody. Do you want 1 microgram per mil or 5 or 10 micrograms per mil on your sample? You usually want to try a range of different concentrations. Also, incubation times. It's not uncommon to do 30 minutes or an hour incubation time with your primary antibody on cells, maybe several hours for tissue or even overnight at 4 degrees. Which is best for your antibody of interest? Often there's recommendations from your commercial source, but you want to test it on your own. Don't just go with what they recommend because every assay is different, every sample is different. And though we validate our antibodies carefully, we can't always predict what your sample will need. And finally, a blocking solution too. I mentioned different blocking solutions. Not every block is the same for every antibody. I won't go into antigen retrieval much, but just realize that if there's a chance that your antigen binding site has been masked, for instance by your fixative, sometimes you can treat your samples with heat or pressure or certain buffer components to unmask those binding sites. There's a lot of different protocols out there for antigen retrieval and some good review papers. Most antibodies don't need it. And sometimes your commercial source will tell you. On the right here you can see an example where I've used too much antibody to label this tubulin sample. And it's caused a loss of resolution and oversaturation using a 20 microgram per mil. This is with our antitubulin, alpha antitubulin detected with a mouse antitubulin, and then a secondary with Alexaflor 555. On the right-hand side, you can see where I've used 5 micrograms per mil, which was right for this sample, and you can see some very good detail there. If your antibody is too old or stored improperly, it can degrade over time, leading to precipitants in the tube or nonspecific binding in your samples. Here in the top, you can see some examples of cells that weren't so great. On the upper left, this was that Golgi image I showed earlier. A lot of speckles off cell, a lot of speckles on cell, nonspecific binding that the blocking couldn't block. If you use a new antibody, in this case anti-Golgi 97, you get nice specific Golgi labeling. On the upper right, you can see a tubulin antibody that's degraded, giving you the sort of pearls on a string appearance to the tubulin. But a new one, you get very nice features like down below it. Remember, quality primaries means you get a quality labeling of your antigen. Very important. And I often have people call in here to tech support asking if their lot is a recent one. Maybe they forgot to put the date on the vial. So it's good to check that. Step 3 in our 5 steps is to detect your label. This is to detect with a secondary, for instance, or an amplification technique, and what controls to use. Secondary antibody usage is a very common technique. There's lots of review papers, again, out there. But how do you optimize it? This is where your unconjugated primary has been detected now with a secondary antibody that has dyes on it. You want to, again, go with a company that you trust, one that has good quality and a good quality control. They've validated it for these methods. Make sure your antibody is relatively new and within a stable period and stored properly. Very importantly, consider the host and target species. For instance, if you have a mouse primary, you want an anti-mouse secondary and a host that matches the serum that you're using, like, for instance, a donkey anti-mouse secondary. Now, if you're combining with other primaries, for instance, you have one rabbit primary and one mouse primary, you want to make sure that your anti-mouse secondary is cross-absorbed against rabbit, so it doesn't cross-label and label your rabbit primary too, and vice versa, that your anti-rabbit is cross-absorbed against mouse, right? So you can usually look into the certificate of analysis for your antibody to know what it's cross-absorbed against, or in the manual for that antibody, or just call us here at tech support. And then, do you want it to be dye conjugated or biotinylated or enzyme conjugated? We'll talk about amplification techniques in a moment. Realize that we have a web resource, too, that you can select an antibody at this URL. Remember, just like with your primary, you have to optimize your secondaries every time. One protocol does not fit every assay. Again, many labs have a certain protocol they like to stick to. Don't count on it. You need to optimize with a new sample and a new secondary, optimize the concentration. Usually for microscopy, we're talking between 1 to 8 micrograms per mil. Incubation time is very important. For cells, it's 30 minutes to an hour. For tissue, it's at least a couple hours, sometimes as much as 4 hours. And you want it to be in a blocking solution, at least some BSA, like 3% BSA, again, to help prevent nonspecific binding. Or our blockade solution is quite good. Remember, just like with primaries, quality secondaries means better detection, and you want to be as specific as possible. Let's talk a minute about amplification techniques. If you're not getting a strong enough signal with a standard secondary detection, you want to amplify it higher, above background. This is particularly important for low-expressing antigens, or maybe rare cell detection in your samples. In the upper left there is streptavidin biotin, or avidin biotin is another one. This is where you have a biotinylated primary or secondary antibody, and now you're detecting it with this hapten, right, a streptavidin that has been conjugated to a dye. When you use a standard secondary, you might get, as I show in the bottom there, maybe 10 to 20 dyes per target. But if you biotinylate and label with a streptavidin, now you're getting 20 to 40 dyes per target, so a much brighter signal. Then, if that's not enough, if you need an even brighter signal, you can go with the tyramide signal amplification, or TSA. This is where your secondary is labeled with a horseradish peroxidase, and then the tyramide reacts with the HRP in the presence of a little bit of hydrogen peroxide, and then binds covalently to tyrosine residues around the site of that HRP. This gives you hundreds of dyes per target, much, much brighter. But there is such a thing as too much amplification. So you want to consider the relative expression of your target. Is it a high-expressing antigen or a low-expressing antigen? Here's some examples. On the top, I have tubulin. Tubulin is a fairly high-expressing antigen in nearly every sample. Using a direct conjugate or a secondary detection like a goat anti-mouse, like in the middle there, you get a pretty good expression of the tubulin in the sample. But if you go with TSA, now you've oversaturated. You lose some resolution, and it's actually too bright. We had to throw a neutral density filter in to take that image. Whereas if you're going with a low-expressing target like alpha-catenin on the bottom, a direct conjugate or a goat anti-mouse secondary was not enough to detect it. You're not getting a strong enough signal over background. But if you use tyramide signal amplification, now you only have a 15-minute exposure and a nice bright signal with good signal to background. Recently, we have released our tyramide super boost kits, which have a poly-HRP. In other words, there's more HRP molecules on the antibody. And that means more reaction with the tyramide to give an even stronger signal. And you can see some examples here of just how strong it is. And because you get such a strong signal from your super boost tyramide kit, now you don't need to use as much primary and secondary antibody to detect it. So you can see some example dilutions here at the top of your primary antibody, and you're able to dilute down to 1 to 600 or even 1 to 1200 and still see specific signals. You definitely can't get that with a standard secondary technique. Remember, amplification gives you greater sensitivity. Let's talk for a minute about controls. And when I talk to researchers, sometimes they don't run all of the necessary controls to rule out possibility of nonspecific binding or nonspecific signal. So I've mentioned already a little bit about this, but let me make a finer point of it. So autofluorescence, right, where your sample has a background autofluorescence, if you don't have a control with no dye, right, no primary, no secondary, you won't know how much of your signal is due to the secondary and how much is due to the autofluorescence. So have a control with no dye whatsoever that you've washed and mounted the same way to see what that background signal is. If it's very high, you'll want to knock that down. Also, have a no primary control. This is where you only use a streptavidin or secondary antibody to detect, but no primary. But you've blocked it the same way. So here you're testing to see if your blocking is appropriate, or if the signal that you're seeing is due to nonspecific binding of your secondary antibody. If so, you'll want to block better, or maybe go with a newer secondary. And then finally, if you're doing multiple antibodies in your section or on your cells, make sure that you use cross-labeling controls. This is where you use the wrong secondary on purpose in a single color experiment. So if you have a mouse and rabbit primary on your sample, then you'll do a control with just the mouse primary and just the anti-rabbit secondary. And hopefully you won't see any binding. But if you do, now you know that you have the possibility of cross-labeling, and vice versa for the other antibodies, an anti-mouse on the rabbit primary to see if there's any binding. Now hopefully all these controls will be negative. You won't see much signal at the exposure time that you need, but it's an important thing to check when you're first starting an assay. Remember, controls gives you confidence in your results. Let's talk about detecting your sample now. We're moving on to the dyes and some of the hardware. It's important, there's a lot of different dyes spanning the visible and far red and infrared wavelengths. How do you choose which dyes to use? Well, first off, you want to match the dyes that you have. You want to match the dyes that you purchase to the filter sets and hardware settings that you have available. Fluorescene filter sets or light cubes are very common in instruments, so you'll probably have a green dye in there like Alexa Fluor 488 or fluorescene or FITSI. That's very common. Your scope might also be configured such that it has a filter set for DAPI in the blue for nuclear labeling, maybe Texas red in the red, or a Psi3 in the red-orange color, or a far red dye like Psi5 or Alexa Fluor 647. Our light cubes, we here at Thermo Fisher Scientific sell EVOS light cubes for use with our EVOS microscopes, which I'll mention again later. And here you can see an example of those light cubes in the lower right, or traditional filter sets in the upper right, and the sort of band passes that you see in this graph. Now realize that you can sometimes have a problem with bleed through. This is where, let's say you have a green fluorescent dye, like Alexa Fluor 488, and an orange-red fluorescent dye, like Alexa Fluor 555, or Psi3. This might be where one dye bleeds through into the filter of the other dye, such that you see a signal. What can you do? Well, up here in the upper right, you see an example of some cells imaged in the green with Alexa Fluor 488. All you should be seeing are punctate spots, which are the peroxisomes that you're labeling. But here you can see some bleed-through from the Alexa Fluor 555 phalloidin. Those F-actin filaments there are not supposed to be there. They're actually bleeding through from the red into the green. So what do you do about this? Well, first off, you can reduce the dye concentration of the offending dye. For instance, in this image, I would want to reduce the concentration or dilution that I use of the Alexa Fluor 555, such that it's not as bright and thus doesn't bleed through as much. Another choice is to use a different dye combination. So instead of Alexa Fluor 555, I would choose a dye that's farther away from the green. For instance, Alexa Fluor 594, which is in the Texas red wavelength, or even a far red dye like Psi5. Or you can choose a different filter set that has a narrower bandwidth. Most people don't have a lot of filter sets to choose from, so that's not as reasonable of an option usually. Or you can use some specialized imaging software to subtract out one image from another, or special instruments. But usually those first two recommendations are the best, reducing dye concentration or choosing dyes that are farther apart. We have a tool online called the SpectraViewer at thermofisher.com/SpectraViewer. And there you can choose numerous dyes and see their excitation and emission wavelengths. You can even pair it up using that tool to display what your excitation and emission wavelengths are on your instrument. And using this tool, you can choose dyes that best fit your filter sets and be able to see just how much bleed through there might be between them, how much they overlap, how much the dyes might bleed through into the filters that you have. It's a very useful tool, and I highly recommend that you go there to check it out. Remember, careful dye choice means that you have clearly separated labels and specific labels. Okay, moving on to step 4 in this webinar. To get a publishable image, you want to protect your signal. You've labeled with a dye, and now you want to make sure that that dye signal is bright and well retained. The most important aspect of that is your mounting medium. So after you've labeled your cells, for instance, what do? Well, usually you put a cover slip on it. Sometimes if you're imaging in a well, that's not the issue. You just want to image in a buffer. But if you're using a microscope, there's a good chance that you're going to put a cover slip on there. And what do you put under the cover slip? That's your mounting medium. For fluorescent samples, it's very important to use an Antifade mounting medium. When you expose your dyes to a very intense light on the microscope, photobleaching will occur. This is where the dye becomes dimmer as you're looking at it. That's because the strong light is degrading the actual structure of that dye, causing it to not fluoresce as well. An antifade mounting medium has antifade components in it that slow that photobleaching. If you want to mount, image, and then discard your slide, then you want a mountant that has a good refractive index right away, like our slow fade diamond and slow fade gold products. They don't firm up, they just stay liquid. But if you wish to archive your samples, if you want to mount them and image them over the course of several days or weeks, then you want a mounting medium that cures or hardens. For instance, our prolonged diamond and prolonged gold products harden to a firm gel. This means that there will be less off rate or slower off rate of the dyes and antibodies on your sample, and just gelling alone slows the movement of reactive oxygen species and such that can contribute to photobleaching. And you want to choose a mountant with a very good refractive index. Refractive index refers to the bending of light as it moves between substrates. For instance, the light goes through your objective, the glass lens, and then it bends slightly in the air, and then bends again in the glass, and then bends again in your antifade mounting medium. So you want something that's closest to glass with a refractive index as close as possible to 1.52, such that there's less bending. All that bending of light means you lose intensity and lose resolution. And usually you want the brightest signal you can get and the best resolution possible. You'll want a mountant that does not quench the dye of interest. For instance, VectaShield will quench far red dyes like Alexafluor 647. So you want to make sure from the company you get the mountant what dyes are best in that mounting medium and which ones are to be avoided. Our prolong and slow fade products work with pretty much any dye and are optimized for our Alexafluor dyes as well. Prolonged diamond even works with fluorescent proteins, unlike most. And then consider whether or not you want a solvent-based mountant like Cytoseal 60 or HistoMount that we sell, or if you want an aqueous-based mountant like our prolonged diamond. Usually the best choice is to go with a aqueous-based. This is where you just move your samples from their last wash directly into the mounting medium without having to dehydrate or move your sample through solvents, as is commonly done with colorimetric dyes like hematoxylin and eosin. Fluorescent dyes usually don't do well with those dehydration techniques, so definitely, if possible, go with an aqueous mounting medium. Here's an example of that photobleaching that I mentioned in this gallery. You can see that in the bottom row is just PBS with glycerol, and this dye, which is fluorescein or Phtisi phalloidin, photobleaches away and becomes dim very quickly. Whereas if you use prolonged gold, or even better, prolonged diamond, you see that you retain your signal over that entire minute. Now, a minute doesn't sound like a lot of time, but actually it's quite long for imaging, and you need time in order to orient your cells and find what you need without the dye just disappearing. So choosing an antifade is important. But what else can you do? Well, you want to reduce the light intensity and the duration that it's on the sample, because the more light there is, the stronger the light, the longer the light, the more photobleaching you have. So you want to be quick about imaging, about setting it up. If you use a traditional microscope, you might even throw in a neutral density filter while you find the cells of interest. Now, if your dye is just so sensitive that photobleaches away really quickly, even in an antifade, then you might want to consider having a second dye, like DAPI, for instance, that you can use to find your cells and set it up, and then you quickly switch over and take the image in the dye that's not as stable. Remember, your hardware settings play a role too. Exposure time. The longer your exposure time, the more photobleaching you'll get. For those who like data, here are some graphs that show photobleaching. These are normalized to their initial intensity, and you can see over time that the intensity decreases or goes down as you see the lines here. Yellow is just PBS, and for many samples that's the worst. But using an antifade mounting medium like our prolonged diamond in green here, you can see that you're able to retain the signal much better over time. We have a selection guide online that you can use to help choose an antifade mountant. You can see the URL here. Remember, using an antifade mountant means you have a more stable signal and a longer lasting signal to give you the best signal to background. Okay, finally, let's talk about the fifth step in this webinar. This has to do with your actual imaging part. When you say get a publishable image, often people think that that has to do just with your actual imaging on the instrument. But as I've pointed out, the first 4 steps here are actually everything before that. What's the point in imaging something that's not already optimized with all those other techniques? Your image isn't going to be publishable unless you're able to do all those optimizations ahead of time. But now let's talk about the instruments and the software and hardware. First off, here is you can see an image of a typical objective lens that's used in a microscope. There's a lot of different aspects in choosing an objective, but generally you want to go with the best possible objective. Usually an apochromatic or S-apo or super apochromatic is your best because it corrects for chromatic aberrations in color and planar aberrations. So it's all about getting an image that is nice and uniform and co-localizes between colors very easily. And of course, you want to consider the magnification. How high do you want to go? Using oil will give you a brighter, more resolved image, but of course, oil has its own issues. I won't talk too much about choosing objectives here beyond that. But what are some things that you need to do about objectives and the lenses in order to get the best, most publishable image? Well, first off, make sure that your slides and cover slips are clean and dust-free before you begin your assay, and that your objectives are regularly cleaned and inspected because you don't want dust or debris or buffer that's been dripped on it to reduce your resolution. And choose your objective lenses wisely, as I mentioned. Also, the light source in your instrument. Most traditional light sources are like a mercury lamp, which you have to align. You have to make sure that your lamp is well aligned and of the right intensity with your system. Or in confocal systems, aligning your lasers. Mercury bulbs do go bad over time, so you have to consider their age and stability. At the bottom, that black and white graph is sort of an example of how the intensity decreases over time, and then you have to replace the lamp, and it's a little unstable at first, then it decreases over time. Our EVOS systems don't have that problem. Also, align your condenser. If you're doing transmitted light imaging, the condenser lens under the stage for an upright scope or over the stage for an inverted scope has to be aligned and have what's called Kohler illumination. That's where you center the condenser and try to get the best illumination. And those lenses have to be clean too. Here's an example of a traditional microscope where in the blue line you see the light, the excitation light for fluorescence travels from the mercury bulb, bounces off the dichroic mirror in the filter cube, and then down through the objective to the sample on the stage. The dye then gives off a wavelength that's higher, like in the green line here, that goes back up through the filter set to your eyes or the camera. In the red is a transmitted light from a tungsten bulb, for instance, that goes up through the condenser, through the sample, and then up to your eyes or the camera. And there's a picture of a Nikon scope at the bottom as an example. However, what we sell here are the EVOS microscopes. EVOS microscopes have a lot of great characteristics about them. One is that, well, first off, there's no eyepieces, right? But also, it has a very short path length for the light, which means that you're going to get a brighter signal. And that's very important for a publishable image. Get a nice, bright signal. And it also uses a light cube. We sell our EVOS light cubes that have the light, an LED lamp built into it. So there's no more aligning of mercury bulbs or having to replace mercury bulbs or discarding the mercury in a safe manner. Now it's built into that filter cube. So you get a nice, even illumination every time without it going bad. It's good for 50,000 hours. And you have built into this light cube the excitation and emission filters for that family of dyes. For instance, a GFP light cube will have those filters for GFP or fluorescein. And you can see the same, this is an inverted microscope, so you can see that it's the opposite of what the scope was I showed before, where all of the fluorescence is down below in the bottom, going up to your sample and back down to the camera. Notice there's no eyepieces, so you're imaging through the monitor. At the top is one of our EVOS FL Auto automated microscopes, and at the bottom there is our brand new FL Auto 2, the newer version of our automated microscope. Here's some examples of some hardware that has not been optimized. On the left is an image of some DAPI labeled nuclei, and you can see that they're dimmer on the upper right compared to the lower left. That's because this was taken on a traditional microscope where the mercury bulb was not aligned properly. And so you get a brighter sample. If you were to move this sample left or right on the stage, you'd see that the field of view is always going to be dimmer in the upper right. So you'd have to realign. And then to the right is an image of an objective lens that has not been properly cleaned. You can see that there's some dust and debris there, which will reduce your resolution and might cause other optical artifacts. And sometimes fibers can be autofluorescent as well. You can also see a little bit of damage to the objective there where it has scraped against the stage. Always got to be careful about that. If you want to learn more about our EVOS microscopes, there's a link for you at thermofisher.com/EVOS. Remember, proper hardware means you get the most efficient detection. Now, as you're taking your image on your microscope, usually you have some sort of imaging software. This software helps you take the image and helps you combine the different colors into one. And there's some very important aspects to keep in mind to get a publishable image. One is that you want to optimize the settings. You want a good exposure time that's not too bright and not too dim. Sometimes you want to adjust the gain setting. This is the voltage to the camera to get a brighter signal, but be careful. Too high of a gain, you can cause some background signal. Too long of an exposure time and you're saturating. Too low and it's too dim. Also, the light intensity matters a lot. With our EVOS systems, you can change the voltage to the LED. Usually you want it pretty high to get the brightest signal possible, but if you're going to photobleach your samples, you can also just reduce the light intensity by adjusting that voltage. On confocal systems, you want to optimize your laser intensity, raster speed, and raster average, and pinhole size. Also, give some thought to the type of file that you're saving. In most imaging software, you can save as TIFF or JPEG or bitmap. You can save as 8-bit or 16-bit. Why would you choose one versus the other? So, for instance, 8-bit images, that's if you're saving color or if you're saving annotations like scale bars, for instance. 16-bit is better for monochrome images. So if you're doing quantitation, 16-bit has more data on it. So you can get more data out of your sample. Remember, though, that 16-bit images won't open on many off-the-shelf software like Photoshop or Windows Image. It'll appear black or dark because it's only reading the first 8-bit images. So to open a 16-bit image, you need to use an image analysis software like our Celeste software or NIH Image J or Fiji. Here's an example of image acquisition software on our EVOS FL Auto 2 instrument. And let's talk about the settings here. So in the lower right, you can see where you can adjust the light, exposure, and gain, but the image is quite dim. But if you increase those settings too much, like exposure time, now you're saturating the signal. You're losing a lot of specific resolution there because the signal's just too bright. With many software like ours, it'll actually tell you when you've oversaturated pixels by coloring it. Here you can see in blue the oversaturated pixels, and you'll know that you'll need to decrease those settings like the exposure time. Here's an optimized picture. I've reduced the exposure and optimized the light settings such that you're able to see nice signal without oversaturating anything. All right, good versus bad images with those settings. Here on the left is an image I took on the FL Auto 2, which is not optimized. At first, you see some signal. You see mitochondria in red, nuclei in blue, and tubulin in the green. But if you look closer, you start to see some problems with it. For instance, there's a little bit of blue haze in the background, and that's because the gain setting was too high in the blue. In the green, it's actually quite dim. So this is because I allowed it to photobleach too much. Now it's very dim. I didn't properly use an antifade. And in the red, I've actually oversaturated it with too long of an exposure time. So if you correct for those things, if you take it before it photobleaches the green, if you reduce the exposure time in red, and reduce the gain setting in blue, now you see a nice resolved image with good signal to background, and the cell's well centered in the middle as well. This is a good image versus bad. Remember, optimizing your settings means you get accurate data and a good signal to background. Very important. So that's your image acquisition software. But once you've taken your image, you often want to analyze it on a different software package, your image analysis software. Here at Thermo Fisher Scientific, we sell our Celeste software, for instance. But there's others out there, like Image J or Fiji or various commercial ones that you can use. And there are some certain characteristics that you need to keep in mind when considering optimized publishable images. Here I've pulled up a 3-color image where I've taken each of 3 colors, blue, green, and red, in monochrome 16-bit images. And I recolorize it using the merge feature here. Here you can see in the lower right the colored version of that with all 3 colors. But there are some things that you can change, and maybe not in a good way. For instance, on the upper part of this image in the red block, you can see that I can adjust the brightness, the contrast, and the gamma. Now, it's okay to do that in some circumstances. Maybe you want to bring out certain features that you want the reader of your paper to focus on, and that's fine. Just realize that you're changing that picture in ways that may not be ethical, so you have to make any changes very judiciously, and I'll talk in a minute about the ethics of imaging. Now, once you've taken your image, how do you measure it, right? A publishable image isn't just about its appearance. It's also about the data that you get out of it. How do you measure that data? Well, here using our Celeste software is an image of some actin-labeled cells using a phalloidin. Well, one way to get the data is using a region of interest. And here you can see some regions I've drawn around some areas that represent bright signal versus a background. On the left there you can see a circle. That's the background that I'm comparing against. And all those other regions I'm measuring the average intensity of to get a value that I can compare between samples. Now, for a sample like this one, a region may not be the best choice because obviously I'm missing a lot of label there by using a region of interest. And so instead I might use a different way to measure using a thresholding technique. Thresholding is where you take your image and you adjust so that you measure over or under a certain intensity. So here I'm thresholding in blue those areas that are colored, and I'm adjusting the thresholding. Now here you can see the blue does not encompass all the specific areas, so now I want to threshold a bit more. And that seems to be pretty good there. Now if you compare from before, you can see that there's some areas that maybe is covered that I don't want to be covered, and a few areas that I'm missing. But if I go too high, now I'm going to start measuring background and black areas and non-specific signal that I probably don't want to measure. Now, one interesting feature about Celeste is that it has a smart thresholding method, and you can actually teach the system what's specific and what's not. So in the pale blue here, you can see the actin is very nicely selected and none of the background. That's a neat feature of Celeste. Now, I won't go into all the features of Celeste software here, but I urge you to learn more at our website. There's a lot of great function in that software package, including like 3D reconstruction or all sorts of different segmentation techniques and counting. So if you want to count cells in an automated fashion. All right, let's talk for a minute about ethics. And this is something that's not actually covered that much by other people out there, so I wanted to really make a point of it. When is it okay to adjust your image and when is it not? Most image acquisition software will allow you to adjust the brightness, contrast, and color. Maybe you can crop it. Maybe you can adjust your raw image and resave it in a certain way. That's okay, but there's some guidelines you need to follow. Never add something to your image that wasn't there in the first place, except maybe some annotations like arrows or scale bars. Never selectively remove something from the image, and don't selectively enhance one area of the image without performing the same manipulation to the whole image. And usually whatever manipulations you do to one image in your series, you'll do to all images in your series. Don't compare two images that have not undergone that same treatment. And don't crop your image in a way that removes valid, relevant data, even if it doesn't support your desired conclusion. I think we all who have experience have known someone who has broken some of these rules, and it's important to call them out on it. And if you do make any changes, it's a good idea to mention it in the captions of your paper. By the way, that cell on the upper right has not been manipulated. Those are nucleoli in the cell labeled with our cytoRNA select green. I always get a laugh out of that picture. Remember, ethical imaging means trustworthy data, and thus publishable data. So that's pretty much everything that I wanted to go over in this webinar. Here's some very important web resources and some sites on our web pages that you can go to learn more about some of these characteristics. The top one is particularly useful for those who are new to fluorescence, our Molecular Probes School of Fluorescence, which has lots of useful techniques and information. The PDF version of this webinar will have these links. And finally, if you have any questions or need help with protocols, please don't hesitate to contact us at technical support. Here's our email address, and you can feel free to ask for me, Jason. Or you can go to your website, wherever you may be in the world, and go to the Contact Us for Support page and click on Technical Support. We have technical support groups all over the world to help you. I hope that you've learned a lot in this presentation about how to get a publishable image, and I'll be happy to take some questions from you now.
Thank you for that presentation, Jason. We want to get right to your questions and input, so here's a reminder as to how audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. If we are unable to get to your question due to time constraints, Thermo Fisher Scientific will reach out to you via email. And our first question is, in what way do the EVOS microscopes differ from traditional microscopes?
Well, when you look at an EVOS microscope, the first thing you notice is there's no eyepieces. You're looking at a screen. All of them use monitors. when I was teaching microscopy to beginning science students, inevitably the hardest problem they had was just learning to look through the eyepieces. And you have to worry about your glasses and the eye width and all that. So don't worry about that with these scopes. A monitor's great. Everything's digital. Also, depending upon which model you choose, and we have 6 different models of microscopes that you can see at thermofisher.com/EVOS, EVOS, right? There are different functionalities. For instance, the EVOS FL and XL allow you to do manual counting by clicking on the screen to count your cells, and you can make movies and time lapses with your images. Or with our EVOS FL Auto and FL Auto 2 system, which is newer and has more features, you can actually do automated imaging. So you can put a plate on there, set it, and then tell it to go and have it image maybe once every 15 minutes or every half hour, whatever, all through the night, especially if you have an on-stage incubator system, which is available as an accessory with those. And so that automated feature is great, and you can do Z-stacks with it, and you can do tiled or stitched images with it. There's a lot of great aspects, and we'll do demos in lab even. So if you're interested in the EVOS systems, you can check out the web page, or we can also help arrange to put you in touch with a representative who might do a demo with you.
Great, thank you. For our next question we have, I've never heard of charge-based background binding, like when you talked about the signal enhancer solution. Do all dyes have this issue?
Well, remember, this is about the charge on the dye, right? So most dyes have a negative charge. For instance, the Alexafluor dyes and fluorescein, and those have a net negative charge which will bind to positive areas on the cells or tissue. But they're all a little bit different in terms of how badly they'll bind. There are a few dyes out there, like Texas Red, for instance, which are zwitterionic, where it has one positive and one negative, so a net neutral charge. But even they can have a little bit of background binding due to charge. So I got to where I was using the signal enhancer solution, which I helped develop, by the way, in R&D, with pretty much every immunocytochemistry and immunohistochemistry protocol I did. It never inhibited binding, and it always seemed to help with pretty much every dye.
Okay. My lab makes its own anti-fade mounting medium. Why not go with our own formulations?
I've helped develop some of the anti-fade mountants here, either directly in R&D or as an advisory. And I can tell you we've tried every possible homebrew remedy that we have found that have been reported by labs or put on web pages. For instance, sometimes people will use Trolox in solutions or N-propyl gallate. But I'm here to tell you that, or Dabco, they're not nearly as good as what we have optimized. I would definitely recommend going with a commercial anti-fade mounting medium like our Prolong and Slow Fade products, because we've done so much validation with a wide range of different dyes and techniques.
Wonderful questions coming in. We have time for just one more. Our lab uses RPE for flow cytometry because it is so bright. Is it also suitable as a dye option for microscopy?
Good question. Well, RPE, or R-phycoerythrin, is very common in flow cytometry because it does have a very strong quantum yield and extinction coefficient. It's a very strong intensity. However, it's not photostable, even with anti-fade mounting mediums. The photobleaching is very bad. So you'll label your sample, you'll put it on your microscope, and the signal will go away very quickly, which is too bad. But you know what? We've got lots of dyes to choose from that are also quite bright, like our Alexafluor dyes and our Dye-Lite dyes. So I would recommend going with those instead.
Wonderful. Looks like we're out of time for questions. I would like to once again thank Jason Kilgore for his presentation. Do you have any final comments?
No, just check out our webpage at thermofisher.com, and feel free to email us if you have any follow-up questions, and we'll be happy to help you. Thank you for attending.
Okay. Thank you again to Jason Kilgore. I would also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand until May of 2018. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward that announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you again next time. Goodbye.